You know, when we think about the brain, our mental picture is usually the magnificent, wrinkly cerebral hemispheres, the part that handles our thoughts, our language, our voluntary movement.
The star of the show.
Exactly. And then far down, we think of the brainstem, sort of the necessary foundation for breathing and heart rate. But right there in the geographical center, wedged between these two giants, is this tiny, often overlooked, but absolutely foundational structure. It's the ultimate control room.
And we are talking about the diencephalon.
Diencephalon.
Our mission today is arguably one of our most ambitious to fully decode this central structure. We've pulled together a dense stack of neuroanatomy sources. We've got anatomical maps, detailed functional descriptions, and some really specific clinical case studies. And we are going to try and extract every single vital piece of knowledge about this central territory.
And when you say central, it really is. I mean, if the entire brain were a global corporation, this little 4 cubic centimeter space, the diencephalon, it would be the most critical operational center.
Oh, absolutely. It's a place where sensory information meets primal emotion, where your internal thermometer is set, and where the entire hormonal system gets its marching orders.
And consciousness, you can't forget that. It's where your consciousness is maintained.
That's the key insight right there. The diencephalon doesn't just pass information. It integrates and it governs. Anatomically, our sources define this region as the caudal, or posterior, part of the prosencephalon.
The prosencephalon being what we call the forebrain.
Exactly, the forebrain. So it sits centrally, and it actually forms the core right around the third ventricle.
Okay, so if you were looking straight down the midline of the brain, the diencephalon is essentially creating the walls and the floor of that central third ventricle space. So its location alone makes it structurally significant.
It has to be. It's wedged between the cerebral hemispheres, which sit rostrally, or towards the front, and the cerebral peduncles, which lead down into the brainstem, caudally, or towards the back. Its central location basically ensures it has to handle communication across virtually the entire nervous system. It's responsible for everything from detailed sensory processing and the state of consciousness to the minute-to-minute control of homeostasis in the entire endocrine system.
That is truly comprehensive. And structurally, how is this complex territory organized for our deep dive? What are the big subdivisions?
Okay, so our sources break the diencephalon down into 4 major divisions.
Yeah.
We have the behemoth, the central relay hub, which is the thalamus.
Right.
Then the tiny but incredibly powerful hypothalamus, which is the master of internal regulation.
The thermostat and the clock.
Exactly. Below the thalamus, we find the motor regulator, the subthalamus, and then finally you have the roof and the pineal gland area, which is the epithalamus.
Okay, let's unpack this. I mean, to conquer this complex territory and really get a handle on its massive functional role, we have to start with the biggest component, the massive gateway that controls everything we perceive. The thalamus.
Grand Central Station?
I think of the thalamus as the brain's grand central station, but maybe even more like a highly efficient, high-security customs office.
I like that.
It's the one place where every single piece of information, whether it's the pressure of your foot, the sound of my voice, or the start of a deep, abstract thought, it has to stop, get sorted, get reviewed, and then catch a connecting train to the correct part of the cerebral cortex. Is that an accurate way to frame its core function?
It's highly accurate,
Yeah.
and the sources really emphasize this gatekeeping role. The thalamus is the major gateway and distribution center for the cerebral cortex. But crucially, and this is the important part, its role is not passive.
Yeah.
It's not just a simple switchboard. It's actively integrating and distributing information involved in virtually all brain functions. Sensory input, motor command execution, limbic functions like emotion, and complex cognitive functions, including attention, memory, and most profoundly, the maintenance of consciousness itself.
So it's the filter. It's the thing that decides what even makes it to the level of conscious perception. When we sleep, for instance, our sensory inputs don't just stop,
No, they're still coming in.
but the thalamus seems to just turn the volume way down.
That's right, it modulates. It's an active modulator. And when it malfunctions, the clinical relevance is immediate and profound. The sources list significant pathologies linked to thalamic dysfunction.
No.
Because it's so central to motor loops, for example, problems here are seen in Parkinson's disease and essential tremor.
And we noted a fascinating clinical nugget in the source material regarding a specific treatment for that.
We did. The thalamus is so central to that motor loop, the basal ganglia to thalamus to cortex loop, that neurosurgeons can use stereotaxic surgery.
Which is this highly precise, targeted procedure.
Incredibly precise. They use it to treat disabling tremors. And they target a tiny spot within the ventral-lateral complex of the thalamus, a nucleus known as the VIM nucleus.
VIM.
Just by disrupting this very precise pathway, they can dramatically alleviate the tremor in disorders like essential tremor. It highlights how much power is packed into such a small, discrete bundle of cells.
Okay, so let's get physically oriented. How large is this central processing unit, and what are its immediate neighbors? What's it sitting next to?
In the context of the diencephalon, it's massive. The thalamus is a large ovoid mass that makes up about 4/5 of the entire thing. We're talking about two large structures, one on either side, flanking that third ventricle.
Okay. And can you describe the boundaries? Since it's ovoid, I'm guessing it has a front and a back that look different.
Absolutely. The anterior pole, which points toward the front of the brain, is relatively narrow. Conversely, the posterior pole is dominated by this huge complex structure known as the pulvinar.
The pulvinar.
This pulvinar is so large, it actually extends back and hangs over the midbrain structures. It's really prominent.
So the back end is the big bulbous part. And what keeps it separated from other structures? What are the walls around it?
Laterally, it's bordered by the internal capsule, which is that superhighway of nerve fibers running between the cortex and the rest of the brain, and the adjacent caudate nucleus. Medially, it forms the lateral wall of the third ventricle. And often, though not always, the two thalamus are connected across the midline by this little bridge of gray matter called the interthalamic adhesion.
Is that connection essential?
Interestingly, no. Its function isn't totally clear, and some people don't even have it.
Yes.
It doesn't seem to be essential for normal thalamic function.
Huh. So it's not a uniform blob. It has an internal structure which has to be crucial for its organization. What's the landmark that defines this internal geography?
The key landmark, and you can see this in any detailed anatomical drawing, is a very distinctive Y-shaped sheet of white matter called the internal medullary lamina.
The internal medullary lamina.
Think of it as the central highway system running right through the thalamus. And this lamina doesn't just divide the thalamus. It physically organizes it into the three major nuclear groups we're about to dive into. The anterior group, the medial group, and the very large lateral group.
And the lamina itself contains critical functional neurons, right? It's not just connective tissue.
That's a great point, yes. The internal medullary lamina itself embeds collections of neurons that we call the intralaminar nuclei. On top of that, surrounding the entire lateral surface outside of the main structures, there's a unique, thin, shell-like layer called the reticular nucleus.
The reticular nucleus.
This thin layer, it's situated between another layer of white matter, the external medullary lamina, and the internal capsule. It is one of the most fascinating parts of the thalamus, and we'll dedicate some real time to it later because its function is entirely different from all the others.
Okay, so we have this fundamental internal structure. Now, when we look at function, the source material makes a really important distinction. Breaking the nuclei down into two classes based on their job description. We have the classic communication relays, and then we have what sound like higher-level editors.
This functional distinction is absolutely fundamental to understanding how the thalamus operates. We call them relay nuclei, or first order, and association nuclei, or higher order.
Let's start with the relay nuclei. They sound like the direct messengers, the ones just passing a note.
They are the high-fidelity transmitters. That's a good way to put it. They receive signals directly from the major ascending pathways, like the auditory pathway from the midbrain, or the sensory pathways carrying pain and touch-up from the spinal cord.
So raw data coming in.
Raw data. Their job is pretty straightforward. Receive the fiber, maintain the integrity of that signal, and project it to a very specific, circumscribed area of the cerebral cortex.
So they are the immediate sensory and motor processing centers. The first stop.
Precisely. The ventral posterior lateral nucleus, or VPL, which relays body sensation, is the perfect example. It takes that raw sensory data and sends it straight up to the somatosensory cortex. They are the initial step in the information loop.
Okay. Now, association nuclei are different. What are they doing if they aren't just carrying new information from the body? What's their input?
This is where it gets really interesting, because the association nuclei are deeply involved in higher integrative cognitive functions. And their input sources are, well, they're inverted. Instead of receiving inputs from the body's ascending tracts, the sources show they receive massive input from the cerebral cortex itself. Specifically from the large pyramidal cells in layer V, the cortex.
Wait, so the relay nuclei are sending body information up to the cortex, but the association nuclei are getting a massive signal from the cortex and then doing what with it?
They process it. They modulate it, and then they project it back to the cortex, usually targeting the superficial layers 9 and 7K. This defines them not as simple messengers, but as high-level moderators. They're participating in ongoing cortical discussions.
That means the thalamus isn't just a pipeline to the cortex. It's a critical component of the cortex's own internal communication loop. It's part of the conversation.
That's the core realization. That is the thing to take away. And this gives us the two fundamental types of thalamocortical loops.
The first order loop is simple transmission. Body to thalamus to cortex. Easy enough.
Right. And the higher order loop is far more complex.
Yeah.
It's cortex to thalamus, and then thalamus back to cortex. This loop involved those big association nuclei like the pulvinar and the mediodorsal nucleus.
And what's the point of that loop?
It allows the thalamus to participate in things like filtering, attention, memory consolidation, and deep cognitive modification. It's thought that these higher order loops actually make up a majority of the thalamic projection fibers.
A majority? So most of the traffic is actually the cortex talking to itself, with the thalamus mediating.
That's what the evidence suggests. It completely reframes the thalamus. It's not just a secretary forwarding calls, it's a cognitive partner. It's performing a kind of kefted of cognitive editing, modifying the information that the cortex is trying to process. It's the mechanism for sustaining and directing focused attention.
Okay, that is a huge conceptual shift. So now that we understand the two major job classes, we need to dive into the specific neighborhoods defined by that Y-shaped internal medullary lamina. Let's start at the front, with the structures involved in emotion and memory.
The anterior thalamic nuclei.
Right, the anterior thalamic nuclei.
These are located at that narrow rostral end of the thalamus, and functionally, they are the primary relay station for the entire limbic system.
The limbic system, our emotional brain.
Exactly. This means they are heavily involved in emotion, learning, and specifically connecting context with memory.
The sources break this down into three components, right?
Yes, the anterodorsal, AD, anteroventral, AV, and anteromedial, AM nuclei. They are principal recipients of inputs from the mammillary bodies, which is a key part of the hypothalamus, and they are functionally tied to the hippocampal formation, the brain's main memory center.
Okay, so if you have a memory that's triggered by, I don't know, a smell or a strong emotional context, these nuclei are likely involved in integrating those two things. How does that memory information physically arrive at the anterior nuclei?
It arrives via the famous mammillothalamic tract, also known as the bundle of Vic-Desir.
That's a name.
It is. This is a crucial pathway linking the hypothalamic mammillary bodies to these anterior nuclei. They also receive inputs from the fornix, which is the major output tract of the hippocampus itself.
That makes perfect sense. The hippocampus builds the memory, the hypothalamus adds that visceral gut-feeling context, and the anterior nuclei act as the funnel.
Exactly.
So where do they send this integrated emotional and memory signal? What's the destination?
Their fibers project primarily and massively to the cingulate gyrus, which is a core component of the prefrontal cortex and what we call the limbic lobe. By linking the deep memory structures of the hippocampus with the higher cognitive and emotional centers of the cingulate gyrus, the anterior nuclei are crucial for integrating memory and emotion with higher cortical functions.
So it affects everything from alertness to spatial awareness?
All of it.
So when a patient suffers memory deficits from damage to this area, maybe due to specific types of vitamin deficiency or a small stroke, it's not because the memories themselves are gone, but because the ability to access or consolidate them via the circuit is impaired.
Exactly. The connection is broken. They break the connection between the memory storage area, the hippocampus, and the conscious processing area, the cingulate cortex. And that creates what we call an amnesic syndrome.
Fascinating. Okay, let's move medially. This seems to be the heavy-duty integration center, where diverse sensory, visceral, and emotional information all get synthesized before it hits the frontal lobe.
Right, the medial thalamic nuclei. This region is dominated by the large and very complex mediodorsal nucleus, or MD, sometimes called DM. It's often considered the primary integrative center for the entire frontal lobes, specifically handling complex executive functions.
Executive functions, so planning, personality.
Planning, judgment, social behavior, the works. And the inputs it receives tell you why.
So what fascinating inputs does the MD nucleus receive?
It's a real melting pot. It receives diverse inputs from the olfactory areas, which helps explain why certain smells can trigger such deep, immediate emotional memories. It also gets signals from the ventral pallidum and the amygdala, the brain's primary emotion and fear center. This combination means it's processing smell, deep visceral information from the body, and pure emotional context all at once.
And its projection map, its output, links straight back to the frontal lobes, which is where we execute behavior and, well, our personality.
Its efforts are enormous. The MD has extensive reciprocal connections with the prefrontal cortex, the orbital cortex, and the anterior temporal areas. This massive connectivity is the foundation for complex cognitive function, effective behavior, and linking our visceral or emotional states to executive decision-making.
That makes the MD nucleus sound absolutely crucial for personality. We know that historical procedures like frontal lobotomy often targeted pathways connected to this very region.
That's a key historical point. Dysfunction or disruption of the MD's connections, either through disease or, yes, historical surgical intervention, is associated with significant psychogenic disorders, shifts in mood regulation, and impaired judgment.
So our personality isn't just in the cortex.
No. It shows that true personality requires the MD to properly mediate and contextualize all these effective and visceral signals before they even reach the higher centers for executive action.
Okay, now we're moving into the lateral group, which, as we established, splits into a dorsal tier, mostly association nuclei, and a ventral tier, the relay nuclei. Let's look at the dorsal tier first. The lateral dorsal, LD, lateral posterior, LP, and the enormous pulvinar, PUL.
And remember, these are primarily association nuclei, so they're heavily involved in contextual awareness, not so much direct sensory transmission.
The LD, the lateral dorsal, it sounds like it overlaps conceptually with the anterior nuclei we just talked about.
It does, functionally.
Oh.
It receives input from the mammillary bodies and the cingulate gyrus,
Yeah.
and then it projects right back to the cingulate gyrus. It functions within the same limbic circuits, likely adding layers of context to emotional memory, sort of refining the work of the anterior group.
Okay. And the LP and pulvinar begin to move us into sensory association.
Right. The lateral posterior, LP nucleus, receives input from the superior colliculus, which is involved in directing your gaze and attention, and the parietal lobe, which is our spatial awareness center.
So the LP is putting together where we are looking with our sense of where we are in space.
That's what it seems to be doing. It's integrating those two streams of information.
We're saving the pulvinar for a full deep dive later, given its size, but generally the dorsal tier is taking partially processed information from other parts of the brain and associating it, rather than just relaying raw input.
Exactly. They are critical for higher-order sensory integration. They're preparing the cortex for complex recognition and planning.
Okay, now for the really highly structured, high-stakes part of the thalamus, the ventral tier. This houses the ventral anterior, VA, ventral lateral, VL, and the ventral posterior, VP complexes. These are the primary relay centers for both motor execution and the body's sensation.
This is where the rubber meets the road.
Yeah.
This is where the motor system talks to the cortex and where the body's signals enter consciousness. Let's start with the motor circuit relay, VA and VL.
So the basal ganglia and the cerebellum are trying to control movement, but they both have to communicate through the thalamus before they can actually reach the cortex, correct?
That's the critical loop. The VA nucleus, ventral anterior, receives major inhibitory signals from the globus pallidus and the substantia nigra.
Key components of the basal ganglia that stop unwanted movement.
Right. They act as a brake. The VA then projects mainly to the premotor cortex, which is area 6. So the VA is handling the initiation and planning of movement, often by acting as that brake, releasing the brake to allow movement to start.
And the VL, the ventral lateral, handles the next stage, the execution.
The VL nucleus also gets basal ganglia input, but critically, it receives massive, powerful input from the cerebellum.
Yeah.
And the cerebellum, of course, is responsible for coordination, balance, and learning skilled movements.
Mm-hmm.
The VL then projects directly to the primary motor cortex areas 4 and 6. This makes the VL the final necessary channel for the cerebellum to influence voluntary motor execution. Is how the cerebellum smooths everything out.
And this helps explain why those surgical targets, like the VIM nucleus, are so effective at treating tremor.
Exactly. The VIM, or ventral intermediate nucleus, which is tucked right into that VL complex, is a major target for deep brain stimulation or thalamotomy.
Why there?
Because tremor is essentially an oscillation, a pathological feedback loop in the motor system, often involving the cerebellum and its thalamic projections. By precisely modulating or disrupting the VIM, surgeons can interrupt this faulty feedback loop, which can restore smooth motor function.
That detail really anchors the clinical significance of these tiny motor maps. It's incredible. Now let's move to the sensory circuit relay. ventral posterior lateral, VPL, and ventral posterior medial, VPM.
These are the somatosensory gatekeepers. All bodily sensation goes through here.
The VPL is the main center for the body, correct? The trunk and the limbs.
It is the body map.
Yeah.
The VPL receives input from the main ascending pathways from the trunk and limbs, the spinothalamic tract,
Carrying crude touch, pain, and temperature.
and the medial lemniscus pathway, which carries fine touch and proprioception, your sense of body position.
So it gets everything.
Yeah.
And how is the map organized? Is it just a jumble?
Oh, not at all. It's highly organized in a strict somatotopic fashion. The sources indicate that a distinct map of the contralateral body is preserved. The trunk is represented anteriorly, and the limbs run posteriorly.
So there's a little person, a homunculus, mapped out in the VPL.
There absolutely is. The VPL projects this detailed, perfectly topographical map directly to the primary somatosensory cortex, SI, which includes areas 3A, 3B, 1, and 2. Think of the VPL as the perfect topographical map of your entire body's surface, just drawn on nerve cells.
So when you feel a mosquito bite on your pinky finger, that very specific piece of information has just traveled along that specific part of the VPL pathway before achieved consciousness.
Precisely. Now, the VPM, the ventral posterior medial nucleus, handles the remaining key sensory inputs.
The face and taste.
That's it. The VPM receives the majority of its input from the trigeminal system, which carries sensation from the entire face, your teeth, your mucous membranes. And it also receives the gustatory or taste fibers.
And where does it send that information?
The VPM projects to the face representation area in the SI cortex and, importantly, to the gustatory cortex for the conscious perception of taste.
That's a fascinating separation of duties. The entire body is mapped perfectly by VPL, and the entire face is mapped perfectly by VPM, which must reflect the immense sensory importance of the head.
It does. And what's intriguing structurally is how the map is inverted. The chin and lower face are represented at the dorsalmost part of the VPM nucleus, and it moves up from there. This incredible minute organization is why damage to a single tiny area of the VPL or VPM can cause a complete sensory loss anesthesia of a corresponding body or face region.
Okay, the thalamus's relay function is so specific that it has evolved two specialized appendages for our highest resolution external senses, sight and sound. We call them the geniculate bodies.
Right. The lateral and medial geniculate nuclei, they're tucked away near the posterior pulvinar, and they demonstrate the absolute peak of thalamic organization.
Let's start with the visual relay. The lateral geniculate nucleus, the LGN.
The LGN is the absolutely essential, non-negotiable relay station for the visual pathway. It receives its input directly from the optic tract, carrying highly structured visual data from the retina. Structurally, it is stunningly precise. It's organized into 6 distinct layers, like a tightly packed stack of pancakes.
6 layers, and this layering is crucial for binocular vision, right? For seeing in 3D.
Absolutely. The input from the 2 eyes is strictly segregated. Layers 1, 4, and 6 receive input from the contralateral eye, the eye on the opposite side of the body. While layers 2, 3, and 5 receive input from the ipsilateral eye, the eye on the same side. This segregation maintains the separation of visual information that you need for depth perception, right up until the cortex can start fusing the 2 images.
And within these 6 layers, the sources delineate 2 specialized functional pathways, often called the M and the P pathways.
Right, this is the functional detail that truly separates the LGN from being just a simple relay. The 6 layers are functionally divided. You have the magnocellular M pathway, which includes the bottom 2 layers, 1 and 2. These neurons are large, they process information very rapidly, and they're crucial for detecting motion, high contrast, and rapid changes in light.
Aware pathway.
Aware and when pathway, exactly. Then you have the parvocellular P pathway, which includes the top 4 layers, 3 through 6. These cells are smaller, more numerous, and they process information more slowly, but with high precision. They are essential for detailed color vision and fine spatial acuity.
The what's pathway. So if you were trying to quickly trap a tennis ball flying across the court, you're using the magnocellular pathway. If you're trying to read fine print or appreciate the colors in a painting, you're using the parvocellular pathway.
That's a perfect application. The LGN ensures that these 2 crucial parallel streams of visual information, the where and the what, are perfectly maintained and directed to the cortex.
And where does all this exquisitely detailed, layered visual information project to?
It projects to the primary visual cortex, which is area 17, also called the calcarine cortex via the geniculocalcarine tract or the optic radiation. And this projection is retinotopic.
Meaning the map is preserved.
The spatial map of the visual field is preserved perfectly, like a photograph, where every pixel corresponds perfectly to a spot in your field of vision. The LGN's organization ensures the cortex receives this highly ordered data, ready for complex image reconstruction.
Okay, let's shift to sound. The medial geniculate nucleus, MGN, the auditory relay.
The MGN is the auditory counterpart, and it's nestled on the ventral side of the pulvinar. It receives its input via the inferior brachium of the inferior colliculus, which is the final major relay in the auditory pathway before you hit the thalamus.
If the LGN is organized by layers for vision, how is the MGN organized for sound frequency?
Well, it doesn't have that strict six-layer structure, but it is organized functionally. It's divided into parvocellular, magnocellular, and medial parts. The critical organization here is the tonotopic.
Tonotopic, meaning sound frequencies are mapped spatially within the nucleus.
Exactly. The sources specify that low-pitched sounds are represented laterally, while high-pitched sounds are represented medially. It's a frequency map.
So just like the VPL has a body map and the LGN has a visual field map, the MGN has a precise frequency map, like the keys on a piano.
Exactly. And it projects from the MGN to the primary auditory cortex. But what makes the MGN more complex than just a simple relay is that while many cells respond strongly to specific frequencies, others exhibit more complex responses. They modulate their activity based on sound duration, intensity, and some even show binaural interaction.
Meaning they're integrating input from both ears.
Right, they're integrating input from both ears before sending the signal onto the cortex. This means the MGN is already helping to locate sound in space. It's doing some of the computational work itself.
We've covered the primary sensory and motor relays. Now we have to turn to the system regulators, the nuclei that handle arousal, consciousness, and filtering. These groups are essential for global brain states, not just for specific tasks.
Right, these groups are crucial for governing when and how all those relays operate. Let's start with the intralaminar nuclei, ILN, those collections of neurons embedded within that Y-shaped internal medullary lamina.
And the two key components there are the centromedian, CM, and parafascicular, PF nuclei.
These are major convergence zones. They receive input from sources that indicate global activity or significant stimuli.
Yeah.
The globus pallidus and the cerebellum, giving them context on motor control. The entire reticular formation of the brainstem, which is the primary arousal system of the brain.
The on switch.
The on switch, exactly. And the spinothalamic tract, which carries diffuse pain and visceral input.
So they are aware of deep pain, motor execution, and overall wakefulness, and they project this integrated arousal signal.
Well,
Where?
widely. That widespread projection is the key. Unlike the VPL, which projects very narrowly to SI, the ILN project diffusely across the entire cerebral cortex. They play a profound role in motor control, sensory-motor integration, and, critically, the maintenance of general arousal and conscious state.
So if they're damaged.
Bilateral lesions here have been linked to apathy, loss of arousal, and memory deficits. It emphasizes their role as the on switch for global cortical engagement.
Okay, now for the truly unique nucleus, the reticular nucleus, RN, that thin shell-like layer surrounding the lateral thalamus, why is this one so special?
The RN is unique because it is the only major thalamic nucleus that does not project fibers to the cerebral cortex.
At all.
Not at all. This immediately signals a specialized internal function. It's not talking outside the thalamus, it's talking inside.
So if it doesn't talk to the cortex, who does it communicate with?
It acts as an internal regulator. It receives input from the cerebral cortex via corticothalamic fibers, and it receives input from the very thalamic nuclei that it surrounds. Its output, however, projects back to those other thalamic nuclei. It never reaches the cortex.
This sounds like a sophisticated feedback loop. The cortex tells the RN what it needs, the thalamus tells the RN what it's trying to send, and the RN then modulates the thalamus itself.
That's precisely its function. It acts as an inhibitory gate or filter, using the neurotransmitter GABA to dampen or modulate the transmission of information through the rest of the thalamus.
So it's the bouncer at the club door.
A very sophisticated bouncer. This function is vital for regulating thalamic activity during shifts in consciousness. For example, during sleep cycles, it helps filter out irrelevant sensory data, which helps maintain a deep sleep state. When you are highly vigilant and focused, the RN ensures only the most relevant inputs are passed along. It is the thalamus's built-in inhibitory noise cancellation system.
We return now to the behemoth at the back, the pulvinar. It's physically the largest single division of the thalamus, dominating that posterior pole. Its sheer size must reflect an incredibly complex, high-level function.
It is massive, and its complexity suggests a pivotal role in advanced cognition that, frankly, we are still fully mapping out. The pulvinar is divided into four main subdivisions: lateral, inferior, medial, and oral.
Given its location and size, what kind of high-level information is it processing? It's not a simple relay.
No, it's a high-order association nucleus. The pulvinar is heavily involved in high-level vision, attention, and sensory association. It gets significant input from the superior colliculus, which is a key midbrain structure for controlling gaze shifts. But more importantly, it is reciprocally connected with vast areas of the cerebral cortex, specifically the visual association areas of the occipital, parietal, and temporal lobes.
So since the LGN handles primary vision, the raw data, what does the pulvinar add to the visual experience?
It adds context, depth, and it directs attention. For example, the inferior pulvinar is heavily connected to the visual association cortex, handling complex pattern recognition. The medial and lateral pulvinar are linked to the parietal lobes, which are critical for spatial awareness, and the temporal lobes, which are involved in object recognition and memory.
So it's putting it all together.
Its main role appears to be in selectively attending to certain stimuli, essentially amplifying or suppressing visual inputs based on what the cortex decides is important at that moment.
It sounds like the pulvinar is a coordinator for the brain's spotlight of attention. If you are searching a crowded room for a specific face, the pulvinar is helping allocate the neural resources to that task.
That's a perfect way to put it. Its sheer size and the density of its cortical connections just underscore its role in advanced cognitive processing that integrates sight, space, and context. It's a very sophisticated piece of hardware.
That concludes our exhaustive tour of the thalamus, the grand central station for consciousness and external sensory input. Now, we have to transition to the hypothalamus, and this is a profound shift. If the thalamus managed the chaos of the external world, we now turn to the tiny structure that manages the internal, silent chaos, the hypothalamus.
It is the undisputed master of homeostasis, the ultimate internal regulator. And despite its small size, we're talking only about 4 cubic centimeters, it is indispensable for survival. It acts as the great integrator, connecting the nervous system directly with the endocrine and autonomic systems.
It controls all the vital involuntary functions: setting your blood pressure, regulating your body temperature, maintaining water balance, governing thirst and hunger, dictating sexual behavior, controlling the timing of your sleep cycle. It's the brain's combined internal thermostat, clock, and energy accountant.
All of that. Anatomically, the hypothalamus forms the floor and the inferior lateral walls of the third ventricle. It extends from the front, the lamina terminalis, back to the mammillary bodies, which are its caudal or posterior boundary.
This small area is just packed with different nuclei. How do anatomists manage to zone this dense population?
It's organized into 2 main categories. You have 3 principal zones running medial to lateral, the paraventricular, medial, and lateral zones, and then 2 major functional divisions running rostral to caudal, the anterior and posterior divisions.
Okay, let's look at that functional split based on position rostral versus posterior, the front versus the back.
The rostral or anterior division contains nuclei like the preoptic, the supraoptic, and the paraventricular nuclei. Functionally, this area is associated primarily with parasympathetic responses.
Rest and digest.
Rest and digest, exactly. It's about cooling the body by initiating sweating and water conservation. If you get chilled, the anterior hypothalamus is inhibited.
And the posterior division handles the opposite.
Precisely. The posterior division contains the mammillary body and the posterior hypothalamic nucleus. This region is predominantly associated with sympathetic functions, the fight or flight response. It increases heart rate, arousal, and heats the body through shivering and constricting blood vessels. The sources really emphasize that these 2 divisions operate in a constant push-pull to maintain thermal and visceral balance.
Okay, so let's get into the hypothalamus's most famous role.
Yeah.
Its direct command over the pituitary gland. Let's start with the posterior pituitary, the neurohypophysis, where the connection is direct and neural.
Right. This connection is facilitated by the magnocellular system. These are large neurosecretory neurons located primarily in the supraoptic nucleus, SON, and the paraventricular nucleus, PVN.
These cells are unique because they are neural cells that function like miniature endocrine glands. They're making and secreting hormones directly.
And they synthesize 2 critical life-sustaining hormones. The first is vasopressin, also known as antidiuretic hormone, or ADH. It's absolutely essential for regulating water balance by telling the kidneys to reabsorb water. The second is oxytocin, which plays roles in social bonding, trust, uterine contraction during labor, and milk ejection.
And their release mechanism is rapid because the axons of these neurons travel directly into the gland itself.
That's the key distinction. The long axons of these magnocellular neurons travel directly down through the infundibulum, that's the pituitary stalk, to terminate in the neurohypophysis, the posterior pituitary. The hormones are packaged into vesicles and released immediately into the systemic circulation in response to neural firing.
So it's a direct, fast neural endocrine connection.
Very fast. And this is why damage to this area can immediately lead to diabetes insipidus, which is a condition where you have an inability to conserve water because you lack ADH. It can be life-threatening very quickly.
Okay, contrast that direct connection with the control over the anterior pituitary, the adenohypophysis, which uses a unique vascular taxi system instead.
Right, this is a whole different system. This is the parvocellular system, involving smaller neurons located mainly in the arcuate nucleus arc and the adjacent periventricular zone. They produce a vast array of chemical signals, releasing hormones and inhibiting hormones.
And instead of traveling down the axon to the gland, these messengers are released into a short, dedicated blood supply.
Yes, a very specialized network known as the hypophyseal portal system. The parvocellular neurons release their hormones, like GnRH, which controls sex hormones, CRH, which controls the stress response, and GHRH, which controls growth. They release them into a capillary network located in the median eminence.
And that blood goes straight to the anterior pituitary.
Those capillaries immediately drain into portal veins that travel directly a very short distance down to the adenohypophysis.
This system means the hypothalamus can control the release of the pituitary's 6 major hormones, ACTH, TSH, FSH, LH, GH, prolactin rapidly, without having to rely on distant systemic circulation.
Right, it creates an extremely high concentration of the releasing or inhibiting factor right where it is needed. For example, the arcuate nucleus produces GnRH and GHRH. The periventricular zone produces somatostatin, which inhibits growth hormone, and TRH, which controls thyroid function. This parvocellular system allows for nuanced, fast-acting control over the body's entire hormonal cascade.
Okay, let's focus on the timing element now. The hypothalamus is the body's clock, and that's primarily run by the suprachiasmatic nucleus, the SCN. We've all experienced the disruption of this system jet lag, shift work. What makes the SCN the master clock?
Its location and its inputs are completely strategic. The SCN is situated right above the optic chiasm, where the optic nerves cross. It is the core mechanism for circadian rhythms, and it functions as the body's primary master clock because it receives direct input from specialized retinal ganglion cells.
From the eye itself.
From the eye itself via the retinohypothalamic tract.
So it's measuring light intensity directly. It's not just about seeing, it's about light detection.
Precisely. These specialized retinal fibers use the neurotransmitter glutamate to signal light intensity to the SCN, and this light signal directly inhibits the SCN's activity. The SCN, in turn, regulates the cyclic release of hormones that control sleep and wakefulness, mainly by setting the schedule for the pineal gland's production of melatonin.
So when the light signal disappears at night,
The SCN becomes active, and it allows melatonin production to ramp up, making you sleepy.
so when you cross time zones, that sense of deep physical disorientation, that is the SCN, attempting to reconcile the new light signal it's receiving with the old internal schedule it's trying to maintain.
That is the SCN in conflict. It uses light input to recalibrate its internal molecular clock, a process which takes time, usually about a day per time zone. And if this tiny clock is damaged, say in experimental models or through disease, the result is completely disorganized sleep-wake cycles. It just demonstrates its critical role in governing our 24-hour cycle.
Beyond the pituitary control, the hypothalamus governs specific behaviors like feeding, emotion, and memory. Let's look at some key players and their deep influence on our day-to-day survival.
First, a structural note on the mammillary bodies. Although they're part of the posterior hypothalamus, they function as part of the limbic system. They get crucial input from the hippocampus via the fornix. Damage here, famously linked to thiamine deficiency and Wernicke-Korsakoff syndrome, results in devastating anterograde amnesia.
The inability to form new memories.
Right. It emphasizes their vital role in consolidating short-term memory into long-term structures.
Okay, now for the centers related to appetite. Back in the 1950s, research famously isolated specific nuclei for hunger and satiety. Where does that balance point lie?
That complex regulation occurs in the medial and lateral hypothalamic zones. Historically, the ventromedial nucleus, VMN, was dubbed the satiety center. If you destroy the VMN in animal models, the animal develops hyperphagia excessive, uncontrolled eating, which leads to massive obesity.
And the opposite.
The lateral hypothalamic zone was called the feeding center. Lesioning this area caused the animals to refuse to eat, resulting in starvation.
Now, we know feeding behavior is far more complex than just two centers, but that opposition remains a crucial principle. What about arousal? The lateral hypothalamic zone also contains the famous hippocretin or orexin neurons.
Right. Orexin or hippocretin is the crucial stay-awake neurotransmitter. These neurons are essential for maintaining wakefulness, arousal, and linking feeding behavior to energy demands. They promote excitement and alertness.
And dysfunction here leads to a well-known disorder.
Yes, the dramatic loss of these orexin neurons is the known cause of the sleep disorder narcolepsy, where patients experience sudden, uncontrollable bouts of sleep.
So narcolepsy is essentially the failure of the hypothalamic wake-up system to function correctly.
Precisely. The neurons that should be firing to keep the brain alert are missing, and that leads to these inappropriate transitions from wakefulness directly into REM sleep.
Speaking of behavior, the dorsomedial nucleus, DMN, is implicated in stress and aggression.
Yes, the DMN is heavily linked to stress responses and coordinating autonomic reactions to threat. The sources note that bilateral lesions or electrical destruction of specific parts of the medial hypothalamus can cause dramatic shifts in affective behavior, sometimes leading to what's called sham rage, or uncontrolled aggressive displays. Again, it illustrates how central this tiny region is to our emotional output.
We've established that the hypothalamus controls everything internal, but it can only do that if it's fully informed. So how does it gather and distribute information globally? What are its major input and output highways?
The afferent, or input connections, are incredibly diverse, which allows it to act as that master integrator of the body's internal status.
Okay, let's detail those critical inputs.
Okay, first you have visceral and gustatory input. It receives chemical and visceral sensory information from the body via the nucleus tractus solitarius in the brainstem. This keeps it constantly informed about internal states like gut fullness, blood gas levels, and basic taste inputs.
It's getting a constant status report from the body.
A constant status report. Second, limbic input. It receives massive connectivity from the entire emotional and memory system input from the hippocampus via the fornix and from the amygdala. This allows it to link memory and emotion directly to physiological reactions, like a pounding heart when you recall a trauma.
Right.
And third, sensory and retinal input. We've already mentioned the direct fibers from the retina to the SCN, plus various other somatic and visceral fibers that give it a complete picture of the external environment as well.
So it knows what's going on internally, externally, and emotionally. Now, where does it send its commands? How does it enforce homeostasis?
Its efferents, its outputs, ensure global control across three major systems. First, back to the limbic system, it projects information back to the thalamus via the mammillothalamic tract, maintaining that critical memory loop, and extensively back to the amygdala and septal area to regulate emotional tone. Second, autonomic control. This is massive. It projects to the brainstem autonomic centers and directly down to the spinal cord preganglionic autonomic neurons. This allows it to directly control the most minute autonomic functions, increasing or decreasing heart rate, constricting blood vessels, changing respiration depth, and governing gut motility. It's direct command and control.
And third is the one we started with.
And third is endocrine control. The entire pituitary control system, both magnocellular and parvocellular, dictates the body's long-term hormonal environment, managing everything from growth to reproduction.
This deep, three-tiered integration sensory input informs the limbic system, which informs the autonomic system. That is what allows the hypothalamus to coordinate an internal state, like increasing heart rate and temperature, with an external complex behavior, like aggression or seeking out a mate. It's the unparalleled coordinator of mind and body.
It really is.
We've covered the master sensory relay, the thalamus, and the master internal regulator, the hypothalamus. We now move to the two smaller but incredibly specific remaining regions, the subthalamus and the epithalamus, which finish off our journey through the diencephalon.
Let's start with the subthalamus. It's located, as the name implies, beneath the thalamus. While it's structurally small, it is functionally crucial. It contains the main orchestrator of motor output outside of the cerebellum, the subthalamic nucleus, or STN.
The STN is functionally tied to the basal ganglia, which is our system for filtering and executing smooth, voluntary movement.
Correct. The STN is a lens-shaped nucleus whose main role is motor modulation. It receives powerful excitatory input from the cerebral cortex, and it sends glutamatergic excitatory output to the globus pallidus.
So what does that mean in simple terms?
In simple terms, the STN acts like a vital accelerator within the basal ganglia network. It promotes movement and helps to refine or stabilize motor output. It's part of the GO pathway.
And if this accelerator runs wild, the results are dramatic, which brings us back to the clinical relevance.
The STN is highly sensitive. Damage here, often caused by a small stroke, can cause a condition called hemibolismus.
Hemibolismus.
This condition is characterized by violent, involuntary flinging and flailing movements of the arm and leg on the contralateral side of the body. The mechanism is fascinating. The loss of the STN's modulating input disrupts the careful inhibitory balance of the basal ganglia, which leads to uncontrolled excitation of the motor pathways.
So that one small excitatory nucleus is the difference between smooth function and violent debilitating motion.
It is a profound example of how critical that balance is. What about the other component mentioned in the subthalamus, the zona incerta, ZI?
The zona incerta.
It's a strip of cells located between the thalamus and the STN. Its function is less defined than the STN, but the ZI is highly connected. It receives input from the motor and sensory cortices and the cerebellum. It's thought to be an important integrative zone for sensor-motor functions, maybe acting as a coordinator between various motor planning systems.
Okay, we move now to the superior and posterior part of the diencephalon, the epithalamus. This area contains the small but complex habenular nuclei and the famous pineal gland.
Let's start with the habenular nuclei medial and lateral. These are sometimes called the brain's anti-reward center.
The anti-reward center.
Yeah. They receive significant input from the limbic system, specifically the septal area, and they project to the midbrain interpeduncular nucleus.
Why the anti-reward name? What does that mean?
Traditionally, they were linked to taste and olfaction, but modern research emphasizes their profound role in regulating the negative side of the reward circuit. They process negative reward signals. Essentially, they signal dissatisfaction, a failure to meet expectations, or an error in prediction.
So they're the disappointment center.
You could call them that. It makes them crucial for emotional regulation, learned aversion, and they are a focus in research concerning depression and addiction, as they can dampen the drive toward pleasurable activities when expectations aren't met.
It's a mechanism that forces us to change our behavior if the outcome isn't good. It ensures we learn from disappointment.
Exactly. Even the smallest parts of the diencephalon are revealed to be these complex regulators of emotion and behavior. Now let's look at the anatomical landmark that has captured imaginations for centuries, the pineal gland.
Right.
The pineal gland is this tiny, unpaired, cone-shaped organ that sits right on the roof of the third ventricle. And historically, Descartes famously called it the principal seat of the soul. He believed it was the point where the mind interacted with the body, precisely because it was the only unpaired structure he could see in the center of the brain. He figured it had to be special.
That historical context adds some color. Now, let's get into the modern scientific structure. What is the gland primarily made of?
The primary cells are the pinealocytes, which are highly modified neuroendocrine cells. These cells, interspersed with glial support cells, are responsible for the gland's main output.
And that output is the famous sleep-regulating hormone, melatonin.
Right. Pinealocytes synthesize melatonin from serotonin. And as we mentioned when we were discussing the SCN, melatonin production is governed by that hypothalamic master clock. Critically, melatonin synthesis is inhibited by light exposure via the SCN pathway, and it dramatically increases in darkness.
This makes the pineal gland the direct endocrine effector of the circadian rhythm. The SCN is the boss, and the pineal gland is the factory floor.
It is the output device. The rhythmic release of melatonin controls our long-term sleep-wake cycles, season biological rhythms, and it even affects reproductive cycles in many species. It demonstrates the hypothalamus's profound influence on external systems through the epithalamus.
And there's also a common clinical marker associated with this tiny gland that's useful in imaging.
Yes, the pineal gland frequently undergoes benign calcification after puberty. It forms these calcified deposits that are sometimes called brain sand.
Brain sand.
Brain sand. And because the gland is located exactly on the midline and becomes visible on X-rays or CT scans, this pineal calcification serves as a highly useful radiographic marker for neuroradiologists to check for shifts in midline structures, say due to trauma or a space-occupying lesion like a tumor.
A built-in landmark. So we've navigated this entire incredibly dense landscape of the diencephalon. We've moved from the high-level cognitive editing of the pulvinar to the minute-by-minute regulation of body temperature in the hypothalamus. What is the overarching synthesis? What's the big picture here?
The diencephalon is the absolutely indispensable nexus that ties together every major functional system of the brain. It is where voluntary motor control, which is mediated by the thalamus and subthalamus, meets involuntary regulatory control, which is managed by the hypothalamus and the epithalamus.
The deep interdependencies here are the most important takeaway. The disruption of a tiny defined area can have these global consequences on behavior or even survival.
Think about the surgical precision we discussed. You disrupt the VIM nucleus, a minuscule part of the thalamus, and you can alleviate essential tremor. You disrupt the excitatory STN, another tiny nucleus, and you trigger violent hemiballistic movements. These are proofs of concept for highly localized, incredibly powerful function.
And the hypothalamus represents the highest stakes of all.
Without a doubt. Damage to key hypothalamic areas causes immediate, life-threatening endocrine or autonomic collapse. Imagine losing the ability to regulate your body temperature, resulting in poikilothermia, where you just take on the temperature of the room. Or losing ADH regulation, leading to rapid fatal dehydration. The sources show that these centers are incredibly specialized and incredibly delicate. The diencephalon truly governs the interaction between our conscious, cognitive experience of the world and our body's fundamental survival mechanisms. It makes us aware of the world, and it keeps the internal engine running smoothly so we can be.
What an incredible deep dive. We started with the thalamus, dissecting its role as a cognitive editor through its higher-order loops, exploring the strict, layered segregation of visual inputs in the LGN, and mapping the sensory relays of the VPL and VPM.
Yeah.
We then moved to the 4cc hypothalamus, exploring the life-sustaining magnocellular system for ADH and oxytocin, and detailing how the SCN acts as the body's master clock, governing everything from jet lag to seasonal changes.
The complexity we've unpacked, from the tonotopic organization in the MGN to the highly inhibitory gating role of the reticular nucleus, it just shows that the diencephalon is so far from being a simple switchboard. It is an active integrator, a moderator, and the crucial physical link between the entire nervous and endocrine systems.
The ultimate lesson here, I think, for anyone tackling complex systems like the brain, is the power of interconnectedness. We cannot isolate the emotional system, governed partly by the anterior nuclei and the habenula, from the body's physical regulation by the autonomic system, controlled by the hypothalamus. It's all one integrated machine, focused on keeping us alive and alert.
And this leads us to a final provocative thought for you to consider and explore on your own time. We've established the hypothalamic role in governing the stress response by releasing CRH, which travels via that portal system to control the pituitary. Given the density of these neuroendocrine fibers and the relentless accelerating pace of modern life and chronic stress, where these fibers are being told to fire continuously, how might this constant subtle bombardment actually reshape the long-term morphology and the hormonal output of these tiny, vital hypothalamic nuclei?
So are we changing the hardware?
That's the question. Are we, through sustained psychological pressure, subtly inducing long-term structural or functional changes in our deepest control centers?
Something to mull over as you navigate your own internal homeostasis. Until the next deep dive, keep learning.